All right, so we have been talking about immune system, right? So we started talking about immune system. We did talk about the main two basics of immune system, which is the innate system, the non-specific system, which is the first and second line of defense, and then the adaptive or specific type of defense system. In terms of the first line, we discussed about skin and mucosa. And in terms of the second line, we spoke about NK cells and inflammation, which is a process, and then phagocytes. So we will pick up from there. We will talk about antimicrobial proteins and fever, and then we will get into adaptive immune system. All right. So all right. So this is where we left off. Who can tell me what did you see in this? What was your find in this homework? Oh. So what was the homework question you spoke to? Like Tom gets a cut on his hand. After several days, he notes swelling, pain, red, and heatness. Heat and redness. Upon opening the vein to relieve the pressure, the presence of pus is noted. So the question that I asked you, that what really happened, what kind of immune response was triggered here, and why there is a pus. Among the processes that we discussed is obviously it's an innate system because we have not even discussed anything in the adaptive. So if it's the innate system, then what it is and why there is a pus. What is the system? So what are the symptoms? What are the clinical symptoms? Swelling, pain, heat, redness. What is this? Inflammation. So therefore, the process is the innate system of inflammation. So that's what is happening here. And why there is pus?
Is it the fifth sign? Like that can come.
That's a fifth sign is like the loose function, but there's a specific thing. If that is present, then only this most likely that causes the pus. Infection, so presence of a microbe, of a bacteria, that might cause a pus. All right. So not only inflammation, there is some infection as well. And then that will actually create pus, might increase the pain. All right. So from here, we will talk about the antimicrobial substances and define their functions, right? So what are these antimicrobial? What is antimicrobial proteins? Antimicrobial proteins are proteins that can enhance the innate defense by attacking the microorganisms directly or by hindering their capability to reproduce. Okay, so either they will attack them or they will stop them to do reproduction. Two ways this is possible. And the two important antimicrobial proteins are interference and complement proteins. All right, interference and complement proteins. So, firstly, interference. What are interference? These are also antimicrobial proteins. And what do they do? They are a family of immune modulating proteins, means antimicrobial, or they actually modify the process of immune system or immune attack system. So what do? Suppose and these kind of proteins, they specifically work on a virus infected cells mostly. All right. So suppose there's a virus that is infecting the cell. Okay, that is infecting the cell. Then what would you see here? The virus is entering, and the first thing any virus upon entering will do, it will start replicating. Okay. So immediately now this particular cell, it will have its interferon gene switched on. The interferon gene will turn on. And that will tell the DNA that, hey, you have a virus, so you need to do something about this. So what it will do, the cell will start producing then in the third step interferon molecules. Okay. Now and that's the host cell number 1 that is already infected by the virus. So now this host cell will leave this cell and will go into the next neighboring cell. It will not be able to do much in its own cell, but it will go into the neighboring cell. Neighboring cell will have the specific receptors, interferon receptors, they will bind there. And once that is like there, that's a trigger point for the neighboring cell to tell its DNA that possible that there is a virus infection coming. So we need to prepare for this. So what the neighboring cell will do, it will start creating antiviral RNA. Okay, antiviral RNA, and in turn that will produce that RNA will produce protein, right? Because RNA to protein is produced, and that protein will take care of if there is a virus infection, then that protein will take care of that. So what interferons do, they actually work on neighboring cells, and specifically when cell is virus infected. Is this process clear? There are two kinds of interference possible, alpha and beta. Okay, sometimes what they can do like interferon alpha or beta, they can also activate the natural killer cells. Who can remind me what these NK cells do? That's innate system. What do they do? How do they attack on cells? What is that? Apoptosis. Yeah. So apoptosis, typically they also work on virus infected cells or cancerous cells. Now, since NK cells are naturally designed to work on virus infected cells, so if interferon can also activate them as well if needed, right? So the next one is complement or complement system. What is this complement? Complement is also a protein. There are 20 approximate blood proteins that circulate in blood in inactive form generally. Okay. It's only required whenever it is required, then only complements are activated. So these proteins are factors like C1 to C9 factor, B, D, P, and their regulatory protein. Okay. They provide major mechanism for destroying foreign substances. Okay. So they activate, enhances inflammation, and also directly destroys bacteria. And if required, they can also take help from the adaptive system as well. So let's see how this works. So complement activation is a cascade of pathways, not just one pathway, it's a cascade of pathway. 3 ways this is possible: classical pathway, lectin pathway, and alternative pathway, right? What is classical pathway? Here, against an antigen, an antibody will be formed, okay? In lectin pathway, lectin is actually a protein that can bind to the surface carbohydrates on a pathogen. So every pathogen or bacteria, virus, anything that they will have, it's a living cell, then it will have a sugar present on its plasma membrane. If you remember for 208, that's called glycocalyx. So lectin actually can bind, lectin is the protein that can bind to those pathogenic surface carbohydrates. It's able to bind that. Lectin is designed for that. It's a protein itself. And what is the alternative pathway? In alternative pathway, there will be series of complement factors which will directly bind to the pathogenic surface. Okay. So 3 pathways possible. First one is the classical pathway. When there is an antigen, the antibody will be formed. Lectin, in this pathway, the lectin itself will bind to the pathogenic organism's carbohydrate on the membrane. And in the third pathway is alternative pathway, when a series of complement factors will bind to the pathogen itself, on the pathogen surface itself. All of these 3 might lead to complement activation. And when I'm saying complement activation, again, it's a cascade of factors that will be activated together. Not just one protein, cascade of factors that will be activated together. So what does it do after getting activated? So it has these 3 problems. So it knows that there might be an antigen, might be a bacteria, might be a pathogen. What will it do? It will act into 3 ways. Firstly, It might recruit an inflammatory cell, which means it will undergo inflammation itself. It might kill the pathogen directly, okay? Or it might do opsonization of pathogens. What is opsonization? This is also a immune system process that uses opsonins, which is a protein, opsonins to tag the foreign invader. It will go and tag the foreign invader so that that can undergo phagocytosis. Make sense? So firstly, it can do inflammation, or it can directly kill, or it can do opsonization, which means it will tag the pathogen so that with the help of a protein called opsonin, so that that pathogen can undergo phagocytosis. Quickly remind me, what are the WBCs that can do phagocytosis? What? What are these? What is the main one? The chief one? Monocyte and another neutrophil. So these are the complement activation pathways, right? Now the last process, this is again a process like inflammation, is fever. What is fever? So we already, obviously, we have all gone through fever, which means it is an abnormally high temperature. And why the temperature it gets high? The reason behind, because bacteria or microorganisms cannot live or above a certain temperature. So that higher temperature actually can kill the microorganism. So therefore, this fever, when we develop fever, especially when we have infection, that actually tries to heal us, right? So what happens when we have an infection? So leukocytes or macrophages, when they are exposed to a foreign substance, which suppose it's a bacteria, then they start secreting a substance called pyrogen. Okay, pyrogen. Pyrogen. Now, pyrogen, what it does, it actually acts on the body's thermostat in hypothalamus. It acts on the body's thermostat in hypothalamus. So hypothalamus is the thermostat of our body. So that whenever we have these pyrogens, like leukocytes and monocytes interacting with microorganisms, it will start creating pyrogen. Whenever we have this pyrogen in our body, then the body temperature will rise. What's the normal body temperature for human beings? And Celsius? 37 degrees Celsius. So then it will rise. Means suppose it will go to 100 degrees, 100 Fahrenheit, which is 40 degrees Celsius, which is well above our normal temperature. So that becomes a problem. Even it can go even further. So more the fever means you'd still have the infection. Now, what is the benefit of doing this whole process? Because this higher temperature causes the liver and spleen to sequester. Sequester means reduce iron and zinc, which the microorganisms require for metabolism to survive. Okay, that's their nutrient. So because liver and spleen both produce iron for blood, so they will sequester that process. So the microorganisms have less amount of nutrients available. Also, it increases metabolic rate, which increases, obviously, if there is some kind of repair required, then it induces that, the repair process. So that's the fever process. Now, question for you. During inflammation, the mast cell releases something that causes vasodilation. What is this chemical? B, histamine, because it's the chemical itself that causes inflammation. Number 5, these proteins are released by the infected cell and help protect surrounding uninfected cells. Say surrounding, so what it will be? C, interferon. What kind of cell they particularly work on? What kind of infected cell? Virus infected cells. What would be the body's response if hypothalamus detects pyrogen? Number 6. What is that? A, increase body's temperature beyond a set point. Set point means beyond our normal body temperature. All right. So the next thing we'll discuss is adaptive defenses. What is adaptive defense? Its adaptive immune system defense is a specific defensive system that eliminates pathogen or abnormal cells in a body, right? So it's the difference between the innate and adaptive is that this is specific defense system. It is not so unspecific anymore. For example, like a pyrogen. Pyrogen detects something and then it raises the body temperature. It doesn't know what exactly it is, it just raise the temperature. If you have a tissue injury or some infection close to your body surface, there could be an inflammation. Okay, so now that process will not exactly know what it is. It will just try to trap the whole thing, keep it localized. Certain times, in although interference or complements are a little bit specific, but still the difference between adaptive and innate is that adaptive is really specific. It knows what it is doing. Will sometime it will trigger some innate responses if needed, but there are two specific cells that is involved in this. So what it does, it amplifies the inflammatory response. Amplify. Remember, it's like if it's there, if it's required, it will make it more. Or if required, it can activate the complement system. Why? Because complement will have the method to destroy the pathogen. So adaptive immune system will set the pathway, will say this is what you need to do, then complement will do that. Okay, but adaptive will be very specifically identified that this is the problem. So this pathway you need to do. Maybe you need to do opsonization, or maybe you need to do inflammation, but it will determine the pathway. Now, the shortcoming is that this needs an initial exposure. For example, like I told you last day, after the two line of defenses fail, then only adaptive defense will actually be activated. So there's a priming time. There's a little time it takes. So characteristics of adaptive, it is specific. It recognizes and targets very specifically the antigens. It is systematic. It is not restricted to the initial site. Like, for example, you had infection in liver, that does not mean it will be only there. Because if the infection have spread, the adaptive immune defense also can spread, because this is done by lymphocytes, and lymphocytes are everywhere in the body. So they are not site specific. Most important thing here, these cells have memory. Okay, so these cells have memory. Means mounts even stronger attack to known antigen. What does it mean? Once it have identified that if the body has this kind of problem, then I need to trigger this particular pathway, it will remember that. If you have a second time exposure, then it will do. That's exactly what vaccines actually do. Once you have that in your body, you have, sometimes if you have taken vaccine, any vaccine, you probably have seen, you get a little bit fever, right? Fever or illness, sleepy, you feel tired, all of that happens because at that point, that's a foreign thing that's being given in your body, which your body did not know before. So initially, your innate response triggers and it thinks that this is something problematic. So therefore, you have a little bit trouble, like either fever or sleepy or whatever. That is depends on the person. For example, I used to have high fever whenever I take vaccine. But after a few days, it will go down. Now, what happens, the body already knows. Okay, so this is the problem. If I'm encountered with this kind of, and typically vaccines are dead viruses. So if there is an actual live virus is there, body will know that I need to do this. So then it will, those cells will remember and will trigger the same response. That's what, this is the very unique thing about adaptive defense system. Okay. Two main branches, humoral, which is the antibody mediated, or cellular, which is the cell mediated. So adaptive immune system is the third line of defense. It attacks particular foreign substances and it takes longer time to react than innate actually. The first one is humoral immunity that is triggered by B cells. The second one is cellular immunity that is triggered by T cells, but both are obviously lymphocytes. So now we'll talk about specifically about the adaptive defense. Let's take 2 minutes break, then we'll pick up from here.